Hi everyone, my name is Elizabeth Morales Rico, and this is my scientific article presentation. The article I chose is titled VEGF, Related Polymorphisms Identified by Genome-Wide Association Study and Risk for Major Depression. The authors include T. Zai, M.G. Stakopoulou, F. Bandes, and others. The paper was published online March 7, 2017. The central question answered in this paper is of whether or not there is a correlation between VEGF and depression. The purpose of the paper is to identify if any specific VEGF-related SNPs are associated with an increased risk for depression. We should care because depression is a common mental illness that affects millions of people of all ages worldwide, and this research can allow for personalized risk prediction for depression. So why did scientists decide to study VEGF? It was because past studies have shown that vascular endothelial growth factor, or VEGF, is implicated in the pathogenesis of depression. Also, VEGF is a multifunctional molecule that has effects on areas of the brain, such as the hippocampus. Something interesting to note is that studies have shown that the hippocampus gets smaller in individuals with recurrent and poorly treated depression. Recently, 4 VEGF single nucleotide polymorphisms, or SNPs, have been linked to depression. They are the following: RS10738760, RS6921438, RS6993770, and RS4416670. The experiment was run on an experimental and a control group. The experimental group had 437 unrelated Spanish Caucasian patients with a depressive disorder. The control group had 477 unrelated Spanish Caucasian patients with no history of depression. The genotyping methods included Quigen blood DNA isolation, competitive allele-specific PCR, or CAS, and FRET-based genotyping systems. The experiment itself had quite a few steps. First, 5 milliliters of blood was drawn from the patients, and then the genomic DNA was isolated with the Quigen blood DNA isolation kit. This then was evaluated for integrity and concentration through 1% agarose electrophoresis and spectrophotometry. After, the 4 SNPs were genotyped using the competitive allele-specific PCR, or CAS chemistry, coupled with the FRET-based genotyping system. Genotyping was confirmed by using an assay based on a combination of multiplex PCR and biochip array hybridization. The central question answered in this paper is of whether or not there's a correlation between VEGF and depression, specifically if any VEGF-related SNPs are associated with an increased risk for depression. Researchers found that among the 4 SNPs studied, only RS4416670 was significantly associated with depression. There was no association with depression with the other 3 SNPs. The tables to the right, 2A and 2B, show that RS4416670 was associated with an increased risk of depression. Minor allele C was also seen to be more frequent in patients with depression as well. In this study, the central question was whether or not VEGF was associated with depression. Researchers found that there is a link between VEGF-related SNPs and an increased risk of depression. This means the individuals carrying this VEGF SNP are more likely to develop depression. This is because there's a significant downregulation of VEGF blood levels during or after exposure to stress, and or these patients have a greater vulnerability to stress. Future studies are needed to show whether or not this specific SNP shows less tolerance to environmental stress and why this SNP is overrepresented among depressive patients. Overall, this study is important because it may predict the susceptibility to depression and could allow for personalized risk prediction and targeted treatment of depression. Thank you.